Okay, so now we'll be discussing how we read trees and interpret them. Um, so, for instance, if our question is what the most recent common ancestor of a species, of a group of species represented in a tree is, how we interpret the tree, which tips are evolutionarily closer to which other tips, why some branches, also called edges, longer than others, why some branches split in two, some in three. So these questions we'll be addressing now. Okay, so tree is basically a set of nodes, so representing species, could also be populations depending on what the tree is showing, nodes and edges or branches. Okay, these are branches, these are nodes, these are internal or interior nodes, and these are exterior or external nodes. And this is the root of a phylogeny. Sometimes some phylogenies don't have roots, and some have roots, rooted, unrooted. It depends. If you have data to construct the root, then you can have a rooted phylogeny, which gives you much more information. If not, then you might have a not unrooted phylogeny. If we had an unrooted, if we hadn't had a root here, then basically the lamprey branch here would just extend like that. The time can flow in either direction, to the right, towards the bottom, or upward. That doesn't matter. It's a matter of taste, and all kinds of trees you can observe, you can find in the literature. Yeah, so this tree and this tree, they're the same thing. They represent the same number of nodes, internal, external, the same number of branches. This, elbow doesn't have any meaning. This direct branch and this elbow branch with elbow, they are the same thing. The groups on trees we call, any biological group we call taxon. A single group will be a taxon, multiple groups would be taxa. This you already know. Nodes and roots and edge or branches we already mentioned. An important point here is, given a phylogeny, a pair of species or a group of species more than two, let's say tetrapods. Tetrapods are here, right? Vertebrates living on land. So we can decide on, given this phylogeny, we can study which species could be an outgroup to tetrapods. And here it's straightforward. All these others could be outgroup to tetrapods, right? Because, for instance, this species, this lineage, loopfin fishes, are equally distant to all tetrapods. Okay, they are not closer to amphibians. Their common ancestor with all these groups is here. Is here, loopfin fishes and tetrapods. So loopfin fishes are not any closer to amphibians than to birds, and they could be an outgroup. However, for instance, if we take mammals and amphibians as a pair of species, then snakes would not be a useful outgroup, because snakes would be closer to mammals, as you see, whereas then amphibians are. But amphibians and mammals, for instance, could have an out. If we're studying amphibians and mammals, we could still use lampreys, or we could use loopfin fishes as outgroup for this pair. Okay, outgroup is a taxon equally distant to all others in question, and understanding which lineages could be outgroups here, depending on which species we are focusing on, is an important thing. You should be able to understand what an outgroup is and identify outgroups when necessary. Sister taxa are just taxa which are closest in a phylogeny. So here, for instance, crocodiles and birds could be sister taxa, and lobefin fishes and tetrapods would also be sister taxa. So all tetrapods together and lobefin fishes, they would be closest groups compared to other marine living organisms. Yeah, and bifurcation term, kind of useful to know. It's separation of a node into two, not more than two. Okay, so here we have a bifurcating tree in that the branches always split in two and not more than two. Okay, so the interior nodes we already mentioned, they represent the common ancestors of the groups in question. So here we have the common ancestor 1, 2, and 3. It is equally distant to all three groups. Here we have common ancestor 5, common ancestor 6, 7, 8, and 9, common ancestor just 8 and 9. When we ask about relationships among organisms, who is closer to whom or what, we don't necessarily, unless it's explicitly stated, we don't refer to the DNA similarity directly or phenotypic similarity directly, but we refer to, given the a certain phylogeny, a certain phylogenetic relationship, which group of species share more recent common ancestry than other species. So if we ask, is 3 closer to 1 or to 4, we check where on the tree is the common ancestor between 3 and 1 here, and 4 and 3 and 4 here. So because 3 and 1 share a more recent common ancestor, this suggests 3 and 1 are more closely related to each other. This is our interpretation, okay? Not that 3 and 1 are more similar necessarily phenotypically or even genetically to each other. You might ask, how could two groups be genetically, if we're comparing two pairs of species, how could a pair be more closely related but genetically more dissimilar? I can answer this question in recitation if you wish, but it can happen. Perhaps happens quite often as well. What would the common ancestors genetically or phenotypically look like? This is also a good question. So we might guess that the common ancestors of 8 and 9 should have common properties of both, common traits of both, and then common ancestor of 6, 7, 8, and 9 would have some commonness across all these guys, but whether the common ancestor would be equally close phenotypically, equally close to all of them, we cannot predict. Genetically, again, we will talk about this, but you can have exceptions. Okay, you can have the common ancestor being more similar to this group than to this group, for instance, and why this, how this can happen, you should be able to address by the end of the course, by the end of the chapter. Okay. Now let's look at these trees. Are they, we already mentioned there are different ways that trees can be represented, and we should be able to understand what the topology is exactly how the topology is represented. So a tree which looks like this and a tree which looks like this, are they telling us the same story? And in fact, they are. So they tell us that 3 and 4 are the most closely related groups, and then 3, 4 are next closely related to 2, and then 2, 1, right? And this tree tells us same story. 3 and 4 are most closely related, then 2, then 1. And just flipping the branches, so the order here for this tree on the x-axis doesn't matter. The branching pattern matters. The order on the x-axis doesn't matter. 3 and 4, this branch bifurcation could be flipped. This could be flipped. This could be flipped. And all the resulting trees would be telling us the same story. Yeah. These are all equivalent. In all these trees, 3 and 4 are together, and then comes 2, and then comes 1. Okay. Just to remind you, we can also have different representations. This is kind of a cool representation that has emerged, I think, more recently. A rooted tree, again, it's telling us the same story, right? 3 and 4 are most closely related, and then then to 2, and then to 1. And here's the root. It's kind of cool. Yeah, so the topology, that is this structure, this branching structure here, indicates the relationships, who shares more recent common ancestry with whom. They don't necessarily show how much change happened, so this is some confusion, a confusion I frequently encounter. For instance, this long branch is sometimes interpreted as having this branch having changed more than this branch, but that is not the case. Branch lengths don't necessarily show change. There are some trees where branch lengths do show change, that is then indicated, but here the tree is just telling us about the division pattern, the split patterns. Phylogenes also don't necessarily show how much speciation happened on a lineage. So here it looks as if 2, 3, and 4, these groups are more diverse, have diversified, but it could be that actually there are more lineages here that were just not included in the phylogeny. So for instance, coming back to the vertebrate case, here it looks as if tetrapods are very diverse, right? With a lot of splits. This is just arbitrary. This is just the choice of the individuals who created the phylogeny. The most diverse group out of all these is, you might guess, who's the most diverse, has most species identified. Ray-finned fishes. Okay, the classical fish group. Those are the most diverse. It's just represented as a single line here. So the phylogeny tells us nothing about how many species there are, unless you might also encounter phylogenies where number of all known species are indicated in usually some kind of simplification or abstraction. But otherwise, don't assume that the number of branches indicate the full number of species. Speciation or split events. Yeah, not all descendant taxa are necessarily represented. So small exercise here. Look at phylogeny of so-called pachyderms, rhinoceri, and hippopotami. And as you see, hippopotami are related to cetaceans, dolphins and whales, and other ruminants and pigs. These are, well, I always confuse. I'm sorry, single hooved or double hooved, whatever. And then we have this other group who are also hooved with the horses and stuff, and then rhinoceri and tapirs. And then we have this more distant group of elephants and manatees, some weird, not dolphins, some weird marine mammal, and hyraxes. Okay, so question is, looking at this phylogeny, what kind of evolution relationships can we infer? Are tapirs closer to hyraxes or to elephants or to pigs? So 3 options: hyraxes, elephants, pigs. And do I have the answer? No. Okay, so this could be a good question for the recitation. Okay, think about that, and perhaps discuss with your friends and try to decide on how whether tapirs are closer to hyraxes, again, elephants, or to pigs, evolutionary. This hyrax, this pig. So yeah, another question you can think about: how many internal nodes would we have in a rooted bifurcating tree if the number of tips is n? If we have n external nodes, n species, or n taxa, how many internal nodes should we have? It should be very simple to calculate. Please go ahead. And okay, so that much for reading trees. We had 2 questions. Don't forget, we will have them as recitation questions and or quiz questions. So both, I can also ask them as quiz questions. So think and discuss. Okay, now we will be talking about a new and important point. So clades, so groups within phylogenies, and monophyletic groups. And here we have mammalian groups, not all of them, of course. We have artiodactyls, canids, felids, part of carnivora, rodents, primates. And as you see, rodents and primates are sister groups here, pretty similar to each other. But it turns out that they share a more recent common ancestor with each other than they share with dogs, for instance, right? Their common ancestor is here, rodents and primates. Our common ancestor with dogs goes somewhat farther back. I think one is like 80 million years, the other one is about 100 million years. But it turns out that when we compare our genomes with those of mice and with those of dogs, we find more homology with dogs than with mice. And the question this time I ask you is, why is that the case? Why are our genomes more similar to dogs than to mice? Okay, but the main point in this part of the chapter is about the structure of different groups. We'll learn a couple of terms. One is polytomy. Polytomy is what we were discussing before. So we said we can have trees where branches bifurcate, but then we can also have trees where branch splits into 3 or more groups. Okay, of course, bifurcating trees, they are nicer, but sometimes we just don't have enough information. We might be uncertain about how the splitting event exactly happened. Sometimes it can also be that split happens in such a way that indeed the 3 lineages we find today, they split at the same time. They all go their own ways, more or less at the same time, and that's why that can be why we do not we cannot create a bifurcating tree. But many times we just lack enough information, and with more information we can, many times we can resolve polytomies. Resolving polytomies meaning determining which of these pairs are more closely related. So this was something systematic discussed for a long time. Are placentals closer to marsupials, or are marsupials closer to monotremes among mammals? And it turns out that placentals and marsupials are more closely related. Okay, so now we have another important term to remember. Clade, or a monophyletic group, is a group within a phylogeny where all the descendants of the group's most common ancestor are in that group, are part of that group, and there are no other members which are, how to say, there are no other members of that group which are left out, or which are left out. In other words, we can say all members more closely related to each other than non-members. Okay, and alternatives are polyphyletic, paraphyletic groups, which are not real clades. Okay, it will be clear when we see some examples. So for instance, this same phylogeny of some mammals, and apparently Pachyderma was a group defined that included hippopotami, rhinoceri, and elephants, that was based on the observation that all three have pretty tough skin. Okay, Pachyderma, derma skin, pachy is, I guess, tough or whatever. So can Pachyderma represent a true clade? Are they a monophyletic group? Again, the definition of monophyletic group was a group that shares common ancestor with each other, they're closest relatives of each other, and there's no other non-group member within that clade. And when we study the phylogeny of the Pachyderma, three lineages, we see that all of them are related to non-Pachyderma mammals, right? So it's obviously not a proper evolutionary classification. This trait must have evolved independently in these lineages. So it's not a monophyletic group. Monophyletic group, again, is a group where all members share recent common ancestry with each other, and there's no non-group member inside that group. For instance, here we see an exception, a polyphyletic group, where different lineages are put together, and they would be, let's say, they would be not monophyletic because they are closely related to other groups. This would be such an example, right, of a polyphyletic group. You can also have a paraphyletic group, which where you have a group which share a common ancestor, but there are some lineages which also share a common ancestor, but are not included traditionally in that group. So that would be a paraphyletic group. And you don't really have to remember these paraphyletic and polyphyletic definitions. That's fine. I think it's a bit too much detail, but you should remember what monophyletic means, and you should be able to recognize a monophyletic group when you encounter that. An accurate evolutionary classification, phylogenetic classification, should only include monophyletic groups, and thus to identify such groups, we need to construct also reliable phylogenies. What may cause paraphyletic or polyphyletic classification? So what caused earlier in the past, when such phylogenes weren't available, what caused people to group together hippopotami and rhinoceri and elephants? well, some common traits, right? So if traits evolve independently, that's something we call convergent evolution, independently in different lineages. Or if some traits reverse, some derived traits are lost in certain lineages, then we might infer that wrongly that those groups are related. So, for instance, in the vertebrate case, by studying the lack of limbs in snakes, we might infer wrongly that they're related to fish, but actually this is just a case of reversal, right? Their ancestors did have ancestors of snakes were reptiles with limbs, and then they just lost their limbs. And likewise, we discussed this case. The warm-bloodedness of mammals and birds might suggest perhaps they share recent common ancestor relative to other lineages, but in reality, given this phylogeny, we can reject that hypothesis, and we can infer that mammals and birds have independently evolved here. Mammals and birds have independently evolved sometime in their own lineages, warm-bloodedness, homoeuthermy. Okay. So this important point, convergent evolution or reversal of traits can cause such classification. A bit of exercise. This is a phylogenetic tree of primates, and our questions are monkeys here, New World monkeys and Old World monkeys. Are monkeys a monophyletic group? And well, you can decide. This can also be a nice quiz question. Are monkeys a monophyletic group? How about hominoids? And I can tell you already, hominoids, yes, are a monophyletic groups. Monophyletic group. These guys are all called hominoid, and they share a common ancestor with each other that is not shared with other lineages. How about reptiles or fish, given the phylogeny, vertebrate phylogeny here? For instance, if you study fish, there are different groups of fish, right? There are these sharks, ray-finned fishes, the classical fish, and also lobed-finned fishes, which have lungs. And here we have tetrapods. So tetrapod, first we can ask, okay, is tetrapod monophyletic group? Was I asking about that? No. Are tetrapods a monophyletic group? Answer is yes, right? They all share a recent common ancestor. A common ancestor. And all the descendants of that common ancestor are tetrapods. How about reptiles? So this is a reptile. Birds are classically, we're taught not to belong to reptiles. So the classical definition of reptile is not a monophyletic group, but if you also include birds, then it would be a monophyletic group. Actually, birds, as we discussed also recently, they're just a branch of dinosaurs, so they're all reptiles. We are different, luckily. How about fish? Okay, so are fish a monophyletic group? And studying the tree, so the common ancestor of all fish lived here, right? But it also has among its descendants all of us. And if we are not included in the fish group, then fish are not a monophyletic group. As you see, lobed-finned fishes are actually closer to us evolutionarily, are closer to us than to ray-finned fishes. We actually, our body plan probably evolved from something like a lobed-finned fish. we'll learn more about that later. Okay. Now we'll talk about roots of trees briefly. So here we have a tree, and as you see, it has no root, and potentially it could have roots here. It could have a root here, perhaps somewhere else. These are different bacteria. And for instance, if the root were here, what it would tell us versus if the root were here, what it would tell us might be somewhat different. So let's ask first how a root is placed, how we can decide where to place the root of a species and of a group of taxa, and that is by using outgroup information. Okay, so if we have an outgroup and the outgroup is, if we added that outgroup to the tree, and if it added here, then we would have the root over here. If it added here, then we would have the root over here. Okay, so that's one thing. We use outgroup information. Two other questions. How many possible roots can we have in an unrooted tree with k branches? And this is a question for you to discuss. Again, it can be a quiz question and or we can discuss during recitation. If we have a tree like this, how many potential roots could such a tree have? Here we have 3 roots, for instance, right? 3 potential sources. It could be here, one could be here, one could be here. What would be the total possible number if there are k number of branches? K total branches. Think of that. And second question is, what does it matter where the root is? Well, okay, let's discuss this using the example we have here from the book. Okay, if we had the root here at A in this branch, we would have 4 and 5 as closely related. So in all these trees, 4 and 5 are closely related, right? Okay. In all these trees, 4 and 5 are closely related, but the trees aren't exactly the same. If we place the root here, then 4 of 5 would be closely related, and then 3, 1, and 2 would be more closely related groups. Whereas if we added the root here, then 4 and 5 and then 3 would be closely related groups, and 1 and 2 would be outside, right? And if we place the root here, then we would have, as shown here, 1 as the first splitting branch, and then 2 would split second, 3 would split, and then 4 and 5 would be together, right? Depending on where you place the tree, the patterning, the branching patterns in the tree will change. So where you place the root is does make a difference. Okay, and so using correct outgroup information to add a root is something of significance. Okay, and let's discuss different types of phylogenies before we finish this session. Okay, first, one most common type of phylogeny that we were also exposed to until now are cladograms. So these only show evolutionary relationships, just the branching pattern. Another type are called phylograms. They show not only the branching pattern, but also the branch lengths represent some measure of evolutionary change. So it could be phenotypic change or DNA sequence change, as we will see soon. And we will also briefly discuss what causes short or long branch lengths. which will be related to our discussion on how come the human genome is closer to the dog genome than to the mouse genome. And a third type of phylogeny is chronogram. So here, the branch lengths show time. So we see time directly on the tree and not evolutionary change. And it can be useful to determine, study speciation times, split times, patterns. We'll see a couple of examples. So here we have a phylogeny. Okay, what do we have here? Human, chimp, bonobo. You were asking about chimps and bonobos, our relationship with them. As we had mentioned, don't forget this point. Bonobos, for instance, cannot be used as outgroup when we compare humans and chimps, but gorillas can because they are equally distant to both humans and chimps. And humans are equally distant to chimps and bonobos, right? And this is important. And here we have a set of anthropoids, old world anthropoid species, or old world monkeys in their own their old name. Baboons and macaques. Here, macaque is, for instance, represented as one lineage, but actually there are many macaques, and some monkeys, some other monkeys, and you also see quite a few polytomies. So here is a polytomy, another polytomy, yet another polytomy with 4 splits. Here as well, 4 splits. So again, this can be because the split times were too close by or not enough data. Here's another phylogeny of what? You tell me. So you see HIV and also SIV. So this virus is human immunodeficiency virus, which you can already see has the HIVs that we carry are sub-branches of SIVs found in other species. So whenever you see, so this should be chimpanzee, SIV from chimpanzee, another HIV strain, SIV from chimpanzee, SIVs from other primates, another HIV, some other SIV from, I forgot what primate species, another SIV. Okay, so here, this tree is interesting. It represents here the branch lengths, represent the amount of change. So here we had a cladogram just representing the branching patterns and nothing else. Here, the branch lengths actually indicates how much change there has happened, and that is based on, how many mutations they accumulated, these strains have accumulated. So we have 2 different sources of information. And so, for instance, when you compare these 2 HIV strains, this suggests that this strain, HIV2B, has changed more than 2A. And that could be because perhaps it got transmitted more often. We can also see things like how many, I might give you this phylogeny and ask you how many times HIV might have infected humans from primates, minimum how many times, and you should be able to answer. And finally, a chronogram describing the evolution of orchids. And you see time here, 0 to 125 million years ago. You have an outgroup species used to root the tree here. And we see that this is first splitting off, and then we have a couple of species which have here, you might imagine, what do these triangles represent? It's already shown, 50 genera. So how many genera, genera is multiple of genus, how many species and genera have evolved in each of these groups? And what you can see is this diversification, or also so-called radiation. Radiation in evolutionary radiation means rapid speciation event of many lineages splitting from each other. This appears to happen closely after, not right after, but closely after the so-called KT boundary, the boundary between the Cretaceous and the, I think, Tertiary. And yeah, here, and this is a description of a number of families across, not just plants, in total found in the fossil record. And this is a period when there's a dip in the number of families, a lot of extinction, that is, and that is linked to also some the deposition of some weird elements in geological layers that are dated to that time. And this actually turns out to be the period when dinosaurs also go extinct. So there's a huge mass extinction, and these orchidae apparently, or how they're called, orchid-related plants, they diversify after this event. So potentially they might be occupying new niches in the after a mass extinction event. Okay, and come back to the question about branch lengths. Why are some branch lengths short versus long? We already answered, so that if there's more change, but then you might ask, why does there happen more change? So for instance, in the case of SIV, aren't two SIV strains on Earth for the same amount of time? Why should one lineage be changing more than another lineage? And I think we also gave a potential answer to that. If one is transmitted more often, it will also be changing more often. So there are two main explanations, but one is much more common explanation. So if a lineage has a shorter generation time compared to other lineages, then this will lead to faster evolution. So for the same time period, it will evolve more rapidly, okay? More rapidly, change more rapidly. Of course, occupying new niches, etc. This can also lead to rapid adaptation, but adaptation is just one aspect of evolution, right? And these trees, they represent overall, for instance, a tree like this is representing here genomic similarity, and not adaptation will only be a small contributing factor to these long branches. Long branches mainly represent just the background process of neutral mutations appearing and fixing, and that will be driven by mainly generation time differences. So for instance, here, this is a tree unrooted, unfortunately. This was pretty early back in the time, but if we root it, we'll also get a similar tree where you see human and chimpanzee and mouse and rat. And mouse and rat have much longer branches than human and chimpanzee. Overall, the common ancestor of mouse rat, the rodent branch is longer, and also their independent branches. And that is mainly because their generation times are much shorter, right? A mice can have, it's the pregnancy time is about, I think it's 90 or 20 days, and then it reaches sexual maturity by 40 days. So basically in 2 months, you can have a new generation of mice if conditions allow. So within a year, you can have 5 generations of mice. So they are 100 times, they can, if again conditions are fine, they can evolve 100 times faster have 100 times more generations per time period than humans and chimpanzees. Mutation rate can also affect branch lengths. Of course, if we're comparing the different, very different virus strains, for instance, mutation rates could have an influence, but usually mutation rate variance is much, much lower than generation time variance or variance driven by, if we're talking about infectious disease, this variation driven by transmission rates. Okay, and now an interesting question from the book. So we have different trees of plants, so phylogenetic trees of plants, which some of which, so in each of these 5 phylogenies, we have both trees and also grass species. So annuals and perennials. Perennials, trees would be perennials, grass are annuals. And okay, study this and try to guess, should the trees be representing the brown branches or the green branches versus the grass? And think of this question in the context of generation time. Which ones have shorter generation time and which ones would be evolving faster? So grass obviously have shorter generation time and they would be evolving faster, and the ones which evolve faster will have longer branches, right? And which ones have long branches here? The green ones, right? So grass must be represented by green, and tree and shrub, apparently they will be representing the brown ones. Okay, so by studying a tree, you can already infer things about the biology of the species represented. A small extra question, how would you compare the rates across these branches? We'll discuss this more in the next chapter, I think, but just to remind ourselves, could we compare all branch lengths using a 2-sample t-test? The green branches and the brown branches using a 2-sample t-test? And the answer is no, because branches within a group are not independent. This is an important point. Related species are, evolutionary related species are, cannot also be treated as statistically unrelated. They are related, right? And so the properties of one species will be found in the properties of another species, so another lineage. So we cannot treat them as independent samples. And we must, what we could do is we could treat the group, so the grass and the trees of each group as a cluster, and we could calculate the median values of each. So we could have 2 median values for each of the 5 large taxa, and then using those 5 pairs of 5 pairs of short and long values, we could run a paired t-test, for instance, or a Wilkinson sign rank test. That could be a useful approach. So important point is if you want to study quantitative properties of phenotypic evolution across phylogenies, you cannot treat the different species as independent if they're related. Okay, and so let's stop there. In the next session, we will be discussing how we can study the distribution of traits on trees and what kind of conclusions we can draw.